All right, here we have the last lesson, lesson 13, which is going to talk about the environmental impact of medications. Here you have your understandings and your applications. So, the synthesis and creation of many medications do have environmental impact. And we want to be able to describe these impacts, and we're also going to be talking about green chemistry. So the first thing we're going to talk about are solvents. So solvents are traditionally volatile organic substances which are toxic to all living organisms. They affect the nervous and respiratory systems, and many, particularly chlorinated solvents such as dichloromethane, affect the liver and kidneys. Many also contribute to photochemical smog by forming secondary pollutants, and also some are greenhouse gases and are contribute towards ozone depletion. Some may also pollute groundwater. Now, solvents um, are quite common in the process of creating a drug. So, because drugs are complex molecules, their synthesis and extraction involves multiple steps, and in each of those steps, different organic solvents are used. They can be toxic and are often left over at the end of synthesis, leading to problems of disposal, right? How do we get rid of these solvents that aren't necessary? It is estimated that up to 80% of the mass of reactants that does not end up in the pharmaceutical product is due to solvents, including water. Disposal of solvents often involves incineration, which it can release toxins into the environment. Overall, solvents are by far the biggest contributor to the emissions of the pharmaceutical industry. So there has to be a way to use these solvents in the drug process, but then get rid of it at the end. So solvents are assessed in the following ways. The first way is toxicity to workers. So, is the solvent carcinogenic, meaning cancer causing or associated with other health issues? The second way is the safety of the process. Is the solvent highly flammable, explosive, or can it cause toxic byproducts? And the third way that needs to be considered is harm to the environment. Whether the solvent will contaminate soil and groundwater, cause ozone depletion, or contribute to greenhouse gas formation when released or burned. So, all these factors need to be considered when a scientist decides to create a drug, what kind of solvent is used. So, here we have some preferred solvents on the left hand side, things like water, ethanol, propanone. And then on the right hand side, we have some undesirable solvents, including that dichloromethane that was previously talked about, diethyl ether, as well as benzene. So, one of the principles of green chemistry is the use of safer solvents and to avoid the use of auxiliaries where possible. Water was one of the preferred solvents, and it is one of the safest. Another example of a solvent is supercritical carbon dioxide, which is CO2 under pressure, can also be used. Another principle of green chemistry is to prevent waste. Some examples include the production of the arthritis drug Celebrex, where scenarios with and without solvent recycling were rigorously compared. So we're going to be talking about green chemistry later on, but it does have a big environmental impact. Another thing we want to consider is nuclear waste. We previously discussed nuclear radioisotopes in medicine, and some of them have half-lives that can last days, like iodine 131, which is used to treat thyroid cancer. And another example, cobalt 60 is used to treat other forms of cancer, and it has a half-life of 5.3 years. So remember, the half-life is the amount of time it takes for half of the material to decay. So these big differences causes some dilemma in how you dispose of this nuclear waste. Another thing that needs to be considered is low versus high level decay. Low level waste has a low activity, meaning not many radioactive nuclei decay each second to produce that ionizing radiation, which is so dangerous, and usually contains isotopes with short half-lives. Right, that radiation is given off for a shorter period of time. High level waste, on the other hand, has a high activity, which means many more radioactive nuclei decay, and usually contains isotopes with longer half-lives, meaning they give off radiation for a longer period of time. So to consider that disposal, there's three possible methods that are considered: dilute and disperse, delay and decay, confine and contain. Now, depending on what type of waste it is, that'll then tell you which way you need to dispose of it. So let's start with a little bit about low-level waste. Includes items that have been contaminated with radioactive material or have been exposed to radioactivity, such as gloves, protective equipment, tools, syringes. They may be stored on site until it has decayed to such an extent that it can be disposed of as ordinary waste, meaning to a landfill site or released into the sewage system. Some low-level waste is incinerated, which reduces its volume considerably and distributes the radioisotopes over a wide area. So this is an example of dilute and disperse. Low-level waste with some higher activity is often buried underground. For example, in individual concrete canisters or concrete-lined vaults, and they may need to be contained underground for up to 500 years, depending on the activity and its half-life. Now let's talk about some high-level waste, which obviously needs to be taken with a different approach. So some examples includes fuel rods that have been used, other materials from nuclear reactors, and the problem with high-level waste is it will remain hazardous to humans and other living things for thousands of years. Some liquid waste could be converted to glass to make storage easier. It's first kept in a storage pool underwater, usually for a minimum of 9 months, but it could be longer. For example, fuel rods are kept for decades, and after they've been cooled, they could be transferred to dry storage casks, and those could be stored in concrete bunkers. Now, this permanent storage, because it takes so long for the radioactive material to decay, is a major problem, and various solutions have been suggested, such as burying the waste deep underground in stable geological areas. But over thousands of years, it is difficult to predict what processes could occur if the radioactive material is released. And there is some argument that there is no suitable solution for the disposal of this high-level waste. We mentioned before that supercritical carbon dioxide, it could be used to contain radioactive ash after it has been incinerated. Now, switching gears, we're going to talk about antibiotic waste. So hospitals often face a particular problem in dealing with antibiotic-resistant bacteria. So these so-called superbugs are bacteria such as methicillin-resistant Staphylococcus aureus, which stands, or MRSA for short, MRSA. They carry several resistant genes and cause infections that are extremely difficult to treat. And we've talked about before that the extensive use of broad spectrum antibiotics, that is, those are used against a wide range of bacteria, has also enabled infections such as Clostridium difficile to thrive. So, as we can see, on the right we have a picture from a colored scanning electron micrograph, which is used to show the bacteria on the microscopic fibers of a wound dressing. And these bacteria are a major problem in hospitals because they are resistant to those many commonly prescribed antibiotics. They can cause wound infections, pneumonia, blood poisoning in vulnerable people, especially those that have recently had surgery. So, getting rid of these antibiotic waste is another problem that hospitals and other medical places have to deal with. We have previously talked about antibiotic resistance, and this arises for different reasons, but bacteria mutate, and so they would normally not account for a very small proportion of the bacteria population, but increased exposure to the antibiotic dramatically increases the number of resistant organisms as it effectively kills off the competition. So the wide use of antibiotics is a good thing to get rid of those harmful bacteria, but it is also an issue when you have this antibiotic resistance. Antibiotics are not just used for humans. In fact, they're used for many different reasons that you can see there, such as pest control in agriculture, different kind of sanitizers and sterilization and culture selection in research and industry. And all of these uses have led to an increase in that antibiotic resistance. We previously mentioned antibiotics in animals, which is where their feedstock is given these antibiotics as a precautionary measure, whereas they're given to prevent the bacteria from infecting the healthy animals. But these antibiotics pass through the animal waste into the soil and water, and so enter the human food chain, where they increase human exposure of bacteria to the antibiotic. So that leads to also some improper drug disposal. Okay? Expired, unused antibiotics are frequently discarded by households and by the medical profession, and this can result in different contaminations of surface, ground, drinking waters. And studies have shown that in some countries, effluent from pharmaceutical production plants can be contaminated with antibiotics. And all of this leads to more antibacterial resistance, which causes more and more problems to the doctors and drug companies when they're trying to prescribe and create new drugs. So this all leads to what has been promoted as green chemistry or sustainable chemistry. Its approach to chemical research and chemical industrial processes that seeks to minimize the production of hazardous substances and their release into the environment. So there are 12 principles of green chemistry, which you can see there. They're going to be going over on the next couple of slides. Each of them are used to promote more environmental friendly methods and using chemicals and using solvents that are more beneficial. Okay, here are some more. A lot of it is using smaller amounts, using just safer chemicals in general, and then preventing pollution, just more environmentally friendly techniques. As we know, the drug design process and the creation of these drugs is a multi step process. So they have to create the best synthetic route, which can involve using readily available materials, having as minimum number of steps as possible, having a good atom economy and good yield of your product whenever possible, using as little solvent and energy as possible during the process. So the atom economy is something that can be used as a measure of how efficient a particular reaction is in terms of converting as much of the starting materials as possible into useful products. So there's a general equation where the atom economy is found by taking the molar mass of the desired product divided by the total molar mass of all of the reactants and then multiplying it by 100%. So this basically gets into how much of your starting material is used to make the product that you are looking for. So obviously the higher percentage you get means more of your starting material has been converted into your product. If it's a low amount, then there is more waste. So here is one example of comparing atom economy to the preparation of a certain chemical, such as 1-phenylethanone. It's used as an enzyme inhibitor, and there are two methods, two ways we can make this chemical. Okay, two different equations which are seen on the screen. And you could see how they are starting with the same starting material, getting the same product, but taking two different methods. So the atom efficiency or atom economy can be worked out. As we can see in method 1, we get an atom economy of about 42%, whereas in method 2, the atom economy is almost 87%. So clearly method 2 is a better method to take your starting material and make your final product, whereas method 1, there's a lot of extra chemicals, extra solvents that are just waste that aren't being used to make your final product. Here's another example of green chemistry, which is in the use of the drug Tamiflu, which is also known as oseltamivir. We talked about this drug as an antiviral that may lessen the spread of the flu virus within the body by preventing the release of new viral particles from their host cells. This drug has attracted particular attention as it is the only orally administered antiviral that may be effective in cases of H5N1 infection, which was the avian flu. Now, the key precursor for the synthesis of Tamiflu is shikimic acid, or its salt, shikimate, okay, which we can see a picture of there below. Now, shikimate is found in low concentration in many plants, but it is found in star anise, okay, or star anise. Okay, it's grown in Vietnam and southwest China, and this particular chemical is found in the star-shaped fruit of the tree and can be extracted in a lengthy chemical process. But the low yields from this process are blamed for the worldwide shortage of Tamiflu in 2005, and again during the flu pandemic of 2009 when many governments stockpiled the drug. So as you can see, if there is none of this material, then it led to shortages and you can't make as much of the drug as you need. So one example is using alternate sources, using this green chemistry to develop another method that is useful to make this drug. So one example is the production of shikimate from fermentation reactions of genetically engineered bacteria. This process has been developed by the pharmaceutical company Roche. Another example is the harvesting of shikimate from the needles of several varieties of pine trees. And even though the yields are quite low, the needles represent a plentiful resource. And a third possible method is the extraction of shikimate from suspension cultures of the Indian sweet gum tree. This is an inexpensive natural source and does not involve genetic manipulation. Okay, this is just one example of how green chemistry and more environmentally friendly methods have been beneficial to the drug industry. Here are some other examples of green chemistry. The production of the drug Viagra uses a modified reaction route that produces just a quarter of the waste. The synthesis of the drug ibuprofen has been altered from a 6-step to a 3-step route. As you can see, the atom economy has increased from 40% to 77%. And synthesis of the drug Lyrica was modified to use a natural reagent of an enzyme with water as the solvent to not use as many other organic materials. And that has eliminated the emissions of more than 3 million tons of carbon dioxide compared with the original process. Okay, so all examples that are now implemented using green chemistry. All right, last but not least, let's talk about just a couple of questions to wrap this up. Number 30 says, describe some of the problems caused by leftover solvents in the pharmaceutical industry. Okay, there are many different examples of these leftover solvents. They could include problems of disposal, including when they incinerated, they release pollutants and greenhouse gases and toxins. Solvents can contaminate groundwater and soil, and solvents can be hazardous to the health of workers. Number 31 says, state some of the sources of low-level radioactive waste arising from nuclear medicine and explain how this must be treated. So protective shoe covers, clothing, gloves, paper towels, contaminated implements. These are all from hospitals and other medical facilities. That's that low-level waste where the nuclear activity is low and there is a shorter half-life. Interim storage in sealed containers for radioactivity to decay before conventional disposal. So we have to let the nuclear material decay into a non-radioactive material first before disposing of it. Okay, 32 says, the very success of antibiotics in fighting disease has led to the widespread emergence of resistant strains, which today threatens their very usefulness. Discuss this statement. Okay, we talked a lot about antibiotics, how they are so good in fighting disease, but they also lead to resistant bacteria. So we also talked about overuse and overexposure. So because this bacteria is exposed to different antibiotics over a long period of time, that has increased the bacteria that is resistant, and therefore some antibiotics are just plainly ineffective if there are multiple resistant strains. Number 33 asks, what is meant by patient compliance in the context of antibiotic prescriptions? We mentioned this in a previous lesson, how patient compliance is really important, and it's so that patients follow medical instructions, especially when completing the full regimen of an antibiotic treatment, and it helps to prevent the spread of antibiotic resistant bacteria. And last but not least, number 34 says, outline the general processes that should be followed to promote green chemistry in the manufacture of drugs. So you can go through that list of green chemistry principles, but in general, they seek to reduce toxic emissions and waste substances in the manufacture of drugs. This includes reduction in the amount of solvent used, the adoption of synthesis pathways with shorter routes, and the replacement of inorganic catalysts with enzymes, and the recycling of waste.